We just got a letter. Kelly says that she needs help with her GN311 homework. Kelly would like to know how to predict the possible gamete types created by a heterozygous individual that has a chromosome of interest with 3 linked loci. Great question, Kelly. We will explain this concept by showing Kelly a reverse mapping problem in blue from Blue's Clues will give us helpful clues along the way. We will base our genetic map off of 3 loci located on chromosome number 3 in the colorful dog species. Locus number 1 controls fur color with magenta dominant to blue. The middle locus controls tail length with long dominant to short, and the third locus controls voice type with bark dominant to howl. The loci for fur color and tail length are 12 centimorgans apart, while the loci for tail length and voice type are 14 centimorgans apart. The coefficient of coincidence value is 0.81. Our first clue is that we must identify a heterozygous individual to base our prediction and genetic map on. The individual must be heterozygous because it would be impossible to know if a recombination event occurred if the alleles that cross over are the same. We can use Blue's friend, Magenta, as our example organism. Magenta's mother is pure breeding with magenta fur, a long tail, and a howl, while Magenta's father is pure breeding for the blue fur, a short tail, and a bark. Since Magenta's parents are pure breeding, any recombination event that occurred during gamete production does not affect the genotype of the chromosome inherited by Magenta. Magenta is heterozygous for all 3 loci of interest because her parents are pure breeding for the opposite allele at each loci. We can now predict the possible gamete types Magenta can produce. Blue has a few clues for us before we begin. The chart represents the possibility of different crossover situations. During prophase 1 of meiosis, crossing over can occur between homologous chromosomes, which allows for the reassortment of linked genes. This means that offspring can have different allele combinations than their parents. There are 4 possible scenarios: no crossover event, a single crossover event at either position 1 or position 2, or a double crossover event. Each event can produce 2 different gametes, so events must be multiplied by a half to find the probability of one specific gamete type. We will now walk you through 2 different examples. A gamete containing the alleles for blue, longtail, and bark requires a double crossover event. The probability of a double crossover event is determined by multiplying the recombination frequency at position 1 by the recombination frequency at position 2, and then multiplying this by the coefficient of coincidence. This number represents the probability of either double crossover event and must be multiplied by 1/2 to specify the gamete. Here's another example. A gamete containing the alleles for magenta, short tail, and bark require a single crossover event at position 1. The probability of this gamete is determined by taking the difference between the recombinant frequency at position 1 and the probability of a double crossover event. Then multiply this number by 1/2 to account for the 2 possible gametes. The probability of all of the gamete types produced by magenta have been included on this slide for reference. For practice, try to solve for the gamete probability yourself and check your math to our answers. Thank you for writing to Blue's Clues for help. We hope this video, along with Blue's insightful clues, helped you understand your GN311 homework.